Good morning students. Welcome to Leela's tutorial. So today's class will discuss about the biological transfection method. So in last class we have discussed the physical transfection and the chemical transfection. So today it is the biological transfection. Again, the transfection is nothing but transfer of the desired gene into the animal cells. Right? So here the biological transfection means we are making use of some organism. So what is that? So here it is classified into two types. Biological transfection is classified into two types. The first one is viral vectors and the second one is non-viral vectors. That means here we are making use of vectors or the carrier molecules. Right? So the viral vectors are used for transfer of the desired gene into the animal cell. If we are making use of viral vectors, we refer that process as transduction. Right? So transduction, it is the viral vector mediated gene transfer process. So here, what type of viral vectors can be used? That is from different viruses, that is adenoviral vectors can be used, oncoretroviral vectors, lentiviral vectors and other different retroviral vectors and all can be used. Right? So coming to the second one, non-viral vectors. So in non-viral vectors, we are not making use of any viral vectors. Right? So apart from these viral vectors, what is the mode of transferring the gene into the animal cell? That is nothing but your physical transfection and the chemical transfection, which we have discussed in the last lectures. Right? Under physical transfection, we have studied in detail the electroporation method, gene gun or the biolistic method, microinjection and laser mediated gene transfer techniques. Right? And under chemical transfection, we have discussed about calcium phosphate precipitation method, liposuction method, membrane fusion method and cationic polymers, which are used to transfer or integrate the gene into the animal cell. Right? So let us see the biological means of transfection by means of viral vectors. So here you can see the procedure. First we are going to take the viral genome. Right? So this is the viral genome and here you have the desired gene of interest. This is the genome from the animal which you have selected. So from that animal you want one specific gene which is having an superior character. Right? So here the desired gene or the characteristic gene which you have selected is this red color region. Right? So here from the whole genome of that animal you want only this desired gene of interest. Okay, so now this desired gene has to be introduced into the viral genome. Is that clear? So here you can see that by means of restriction endonucleases, you cut these desired fragment of DNA and introduce that into the viral genome. Okay, so here you can see this is the viral DNA. The black color one represents the viral DNA and the red color one is the desired DNA of interest. Is that clear? So here we have shown as a DNA model, right? Whereas here I have marked as strands. Okay, but here in this viral genome you have excised this region and here also by means of restriction endonucleases, and this region, the desired gene or the desired fragment is introduced here in the viral DNA. So now this is called as recombinant DNA or the modified DNA, where it consists of the viral genome and also the desired gene of interest from another organism. Is that clear? So here now it is called as the modified DNA. This modified DNA will be introduced into the virus. So now here the virus is called as vector. That means the virus that carries the desired gene of interest in its genome is referred as vector. Right? So now this virus acts as a carrier vehicle for this desired gene of interest. So virus helps us to transfer or integrate this desired gene into the animal cell. right? So it is called as the vector. The virus containing or virus carrying the desired gene of interest in its genome is referred as vector. Is that clear? Or virus containing the modified, carrying the modified gene is referred as vector. Now the vectors, the viruses are allowed to divide, right? So then they are added to the animal cell cultures. So now here you can see a single cell, right? I have shown only the single cell and the single vector. Okay, so here in this single cell, now this vector carrying the desired gene will go and attack the animal cell, right? So it is, after that it penetrates into the cell, into the cytoplasm. So now this green dots is nothing but the cytoplasm. The virus or the vector carrying the desired DNA has entered into the animal cell and forms an vesicle around it, right? So the membrane present around this vector is nothing but the vesicle. Okay, so now this vesicle carries this vector to the nucleus of the animal cell. Is that clear? So now this is the vector which is carrying the desired gene, its genome, right? And now the vector, when it enters into the cell, it forms a vesicle. So now this vesicle, right, a membrane bounded or imprisoning the vector will carry this to the nucleus, right? So now you call this as vesicle. So now this vesicle is moving towards the nucleus in the cytoplasm. So here you can see, as it reaches the nucleus inside the animal cell, as it reaches the nucleus, here you can see there is breakage of the vesicle. The vesicle or the membrane breakage is seen here. So as the membrane breaks, there is release of this vector. The released vector will go and bind to the nucleus and allows the genome or allows its genome to enter into the nucleus and get integrated into the genome of the animal cell. So finally you can see the animal cell consists of the desired gene of interest in its genome, right? Now this cell will have ability to express or produce the protein responsible for that particular gene. So here this is the biological transfection method. So transfection, why you are doing the transfection? In order to modify the animal cell to produce some useful product. Is that clear? So it is used for producing certain products, and also in the therapeutic applications are there, where it is curing the cancers, heart defects and gastrointestinal tract defects and all. Okay, so with this we are done with the transfection topic. If you have any doubts, please put it in comments and subscribe for further lectures.